I've been working with the same research group for about 3 years now. I first started in this group when I was doing my master's thesis, before getting a part-time job as a student researcher. This then turned into a 1-year position as a full-time research engineer. And now that I have my own company, as a contracted project manager and occasional research scientist. Academic jobs are, by their very nature, typically short-term and incredibly volatile. Thankfully, I found a workaround. Because of my business experience in the US, I was able to create my own company here in Sweden, and my research group contracts my hours as a third-party vendor. Thankfully, this means that I don't have to bounce from city to city for the first few years of my career, but it is something that those who are looking to get into the biomedical sciences and research in general should keep in mind. It also means that I get to keep using the free coffee at the lab. Ignore me, I'm getting B-roll. Yeah, I'm trying to get back into the YouTubes, but movies when they'll cut from like a super dramatic scene and then they'll just show you like random cars passing on the city or someone pouring their coffee, and it's a pain in the butt. Fruit. Esther! Okay. Welcome to the lab. This is BKVII, that stands for Biomedical and Clinical Sciences, Inflammation and Infection. That's the division that I work for here at Linköping University. Technically, I work for the CBCS, which is the Chemical Biology Consortium of Sweden, but I do so in the Linköping University node, which means this is where I am. So this is my office for today. I'm going to be sharing it with every student in the known universe. You guys check out that view. As a consulting project manager for the CBCS, a lot of the things that I'm responsible for are administrative, which means that the vast majority of the time, I work from home. And seeing as how that's not particularly interesting, today you're coming to the lab with me. So the main reason I'm coming in today is because we're actually starting an experiment with some cells that we started culturing last week. So those cells have been growing and maturing and evolving, and now we get to scoop them up and infect them with tuberculosis to start a new experiment. So today is kind of a weird day. So I have one experiment ending, one experiment that is just starting, and we also have a bunch of admin stuff that I'm not able to do remotely and have to be on site. The biggest of which being an inventory. I know, not super sexy or exciting, but it is really important to make sure that we have all of the things that we need for our experiments, because shipping for some of these things can take several weeks. So we need to make sure that we have what we need for each experiment long before it starts. So today is one of the rare days I actually have to go to the lab. So I actually have to get started on some things, so meeting time. Okay, so cell counter and the lymphocytes. Sorry, not you.
Select your cell population, but it's just
So you were saying that the Countess 2 has a cell count option, a size differentiation option?
so
But if it was a macrophage heavy sample that time, that would have meant it was
yeah.
the same. That's been our ongoing struggle, is
Yeah.
we can't identify which donors are going to have high lymphocyte presence and which donors are going to have lymphocyte proliferation. There's a paper out, this is the theory we've been going with. It was by Cecilia from the La Jolla Institute. It was about T-cell monocyte adhesion during isolation stages as a marker for immune perturbation. So that's our working theory. So either your protocol is better or they're not adhered, they're just sort of there.
Yeah.
I don't know. Either way. Okay, so today we need to harvest some cells that we have already been growing over the past week. So I need to get everything set up so that we can collect the cells that we've been growing, bring them downstairs, and then intentionally infect them with diseases to see how quickly they die. That's the plan. Apparently, I am an incredibly unreliable narrator in the first person because that was an awful summary. In our research group, we mostly work with human primary cells. That means cells that have been collected directly from donors. So anytime you donate blood, Thank you. These cells have been growing in flasks in our incubator over the last 7 days, and if you ever want me to explain more about how we isolate and culture specific immune cells, let me know, because I can definitely nerd out on that. We've been working on developing a number of SOPs or standard operating protocols here in our lab, but there are still a few things that vary from one protocol to the next. One of those being how we harvest them. This week I'm focusing on testing the 2 main methods that we have been using in order to collect our cells. Once the flasks have been removed from the incubator, I remove the medium inside and replace it with one of 2 different reagents, either a FACS buffer or trypsin. Now typically I've been using trypsin, which dissolves the proteins that the cells use to stick to the bottom of the flask and then makes them unsticky and easier to remove. This is fast, efficient, and gives us a large number of cells. However, trypsin can be a little harsh on the cells. The other method is something called a FACS buffer, which is typically gentler, but can take anywhere from 20 to 40 minutes to fully unadhere the cells. Long lab days tend to lead to mistakes, so any procedure that can allow us to generate the same high-quality results while saving time and labor hours is definitely the way we want to go. Okay, so I've got the first round of samples in the centrifuge. The second round of samples is cooking in the incubator with the FACS buffer. And then once these guys come out of the centrifuge, what I'm going to go ahead and do is put them into specialized chambers, add trypan blue in order to tell which ones are dead or not, and then count them with a little machine next door. So, because this is a cell biology lab, there's only so much I can show you. So if I can't show you something, I'll try and find some stock footage to fill in the gap. But essentially what we're doing today is harvesting cells that we've been growing, and then we're going to put them on plates and image them over the next 7 days. We do this with a specialized piece of equipment that is called a live cell image analysis system, and specifically the one that we use is the IncuSite from Sartorius. Live cell image analysis is really freaking cool. It's essentially time-lapse photography, but for a microscope. So you can see how infections develop in real time and how the cells respond to those infections. I freaking love live cell image analysis, so if you guys ever want me to talk more about that, please let me know, because I will rant inconsolably into my laptop for an hour about how cool this stuff is. But in the meantime, I have cells spinning, so I gotta get back to it. This part is optional, but while my cells are spinning, I like to take the culture flasks and visually inspect them with the microscope so that I can be sure that I got all of the cells that I wanted before I throw them in the bin. I know this next part looks a little intimidating to new-time lab scientists, but once your samples are spun down, you want to immediately discard all of the liquid on top of the tube, and the easiest and fastest way to do that is to just dump it. Add a small amount back into the tube before pipetting it up and down gently, and redistribute the cells into the new medium. Once you've done this for all of your samples and you have your cells evenly distributed, it's counting time. Now there are lots of ways to do this, and before now we've been doing it manually with a microscope and Burker chambers, but recently we've been messing around with this Count as 2 system, which has been really nice thus far, but we're still fine-tuning the process. Once I have the cells counted, I'm able to hand the tubes off to my fellow researchers for infection down in the Biosafety Level 3 lab, so that I can get back to the important stuff. Cleaning. I wish someone had told me when I started my biomedical masters how much of my time was going to involve routine cleaning. The last steps are always to make sure we give everything a really good wipe down, shut down our cell culture hood, and of course, take out the trash. But now, it's lunchtime. So the piece of equipment that I need in order to do my experiments is still not working. So we're working on that, but until we get it fixed, I can't start the next project, which means that instead of science, I'm going to go count bottles of media to make sure that we have enough and order more. Also, hi, Alan.
Hello.
I first got into project management at the lab because of my background in business. There's a lot more to keeping a lab running on a day-to-day basis than just doing experiments. Making sure we have the materials needed to do those experiments is just another small part of that. Coordinating labor hours and manpower, scheduling experiments around other research groups, managing our digital storage. There's a lot that goes on in the background of laboratories that most people aren't aware of. And yes, some of that includes counting pipettes in a dark closet. Some of it means learning more about digital management systems. And some of it means taking out the trash. Science is a process, and it is one with a million moving parts. So while a lot of these tasks may not necessarily be what people think of when they get into studying things like biomedical sciences or biotechnology, they are the things that make this lab run. There are plenty more things that I could do today. But you know what? It is almost 5 o'clock. It has been a long day. And yeah, I think I'm done. Yeah.